Hello, welcome to Biology, the scientific study of life. Rapid reproduction, mutation, and genetic recombination contribute to the genetic diversity of bacteria. By studying the mechanisms by which viruses are replicated, researchers also learn about the mechanisms that regulate DNA replication and gene expression in cells. Bacteria are equally valuable as microbial models in genetics research, but for different reasons. As prokaryotic cells, bacteria allow researchers to investigate molecular genetics in the simplest true organisms. Information about numerous bacteria species has accumulated recently with the advent of large-scale genome sequencing. However, except where otherwise noted, we will focus on what has been learned from the well-studied intestinal bacterium Escherichia coli, sometimes called the laboratory rate of molecular biology. The bacterial genome and its replication. The main component of the genome in most bacteria is one double-stranded circular DNA molecule that is associated with a small amount of protein. Although we will refer to this structure as the bacterial chromosome, it is very different from eukaryotic chromosomes, which have linear DNA molecules associated with a large amount of protein. In E. coli, the chromosomal DNA consists of about 4.6 million nucleotide pair, representing about 4,400 genes. This is 100 times more DNA than is found in a typical virus, but only about 1,000th as much DNA as in a human somatic cell. Still, this is a lot of DNA to be packaged in such a small container. Stretched out, the DNA of an E. coli cell would measure about a millimeter in length, 500 times longer than the cell. Within a bacterium, however, certain proteins cause the chromosome to tightly coil and supercoil, densely packing it so that it fills only part of the cell. This dense regions of DNA, called the nucleoid, is not bounded by membrane like the nucleus of eukaryotic cell. In addition to the chromosome, many bacteria also have plasmids, much smaller circles of DNA. Each plasmid has only a small number of genes, from just a few to several dozen. We will learn more about plasmids later in this section. Bacterial cells divide by binary fission, which is preceded by replication of the bacterial chromosome. Notice this picture figure. From a single region of replication, DNA synthesis progresses in both directions around the circular chromosome. Notice that bacteria can proliferate very rapidly in a favorable environment, whether in a natural habitat or in a laboratory culture. For example, E. coli growing under optimal conditions can divide every 20 minutes. A laboratory culture started within a single cell on an agar plate containing nutrients can produce a mass or colony of 10 to the power 7 to 10 to the power 8 bacteria within 12 hours. Reproductive rates in the organism's natural habitat, the large intestine colon of mammals, can be much lower. On doubling of cell number in the human colon takes about 12 hours, for instance, but this is enough to replace the 2 by 10 to the power 10 bacteria lost each day in feces. Mutation and genetic recombination as source of genetic variation. Binary fission is an asexual process. The production of offspring from a single parent. Thus, most of the bacteria in a colony are genetically identical to the parent cell. Mutation, however, can cause some of the offspring to differ slightly in genetic makeup. The probability of a spontaneous mutation occurring in a given E. coli gene averages about 1 by 10 to the power minus 7 per cell division, only 1 in 10 million. But among the 2 by 2 to the power 10 new E. coli cells that arise each day in a single human colon, there will be approximately between 2 by 10 to the power 10 also between two brackets 1 by 10 to the power minus 7 equal 2000 bacteria that have a mutation in that gene. The total number of mutations when all 4300 E. coli genes are considered is about 4,300 by 2000 equal 9 million per day per human host. The important point is that new mutations, though individuals rare, can significantly increase genetic diversity when reproductive rates are very high because of short generation spans. This diversity in turn affects the evolution of bacterial populations. Bacterial populations composed of individuals that are genetically well equipped for the local environment will reproduce more prolifically than populations with less fit individuals. In contrast, new mutations make a relatively small contribution to genetic variation in a population of slowly reproducing organisms, such as humans. Most of the heritable variation we observe in a human population is due not to the creation of novel alleles by new mutations, but do not the creation of novel alleles by new mutations, but to the recombination of existing alleles during sexual reproduction. Notice this figure. Even in bacteria, where new mutations are a major source of individual variation, additional diversity arises from genetic recombination, defined as the combining of DNA from two sources. In most of the cases we will discuss here, the two sources of DNA are the genomes of two separate bacterial cells with genomic DNA from one cell ending up in the genome of the other cell. Notice this figure shows one type of experiment providing evidence that genetic recombination occurs in bacteria. This experiment made use of two mutant E. coli strains, each enable to synthesize a required amino acid, either tryptophan or arginine. As a result, the mutant strains were unable to grow on a minimal medium containing only glucose as a source of organic carbon and salts. When bacteria from the two strains were incubated together, however, cells emerged that could grow on minimal medium, indicating that they made both tryptophan and arginine. The number of such cells far exceeded what could be accounted for by mutation. Most of the cells that could synthesize both amino acids must have acquired one or more genes from the other strains, presumably by genetic recombination. Back to this replication of bacterial chromosome represents. Notice also from one origin, DNA replication progresses in both bacteria or in both directions. Around the circular chromosome, notice that two directions, boom. Around the circular chromosome until the entire chromosome has been reproduced. Enzymes that cut, twirl, red arrow, and reseal the double helix prevent the DNA from tangling. Keep in mind that although the overall direction of DNA replication is outward from the origin in both directions, one new strand at each replication fork is made discontinuously in the direction back toward the origin. Also, notice the inquiry: can a bacteria acquire genes from another bacterial cell? Experiment research had two mutant strains, one that could make arginine but not tryptophan, notice that one, and that could make tryptophan but not arginine. Each mutant strains are a mixture of both strains, which grow a liquid medium containing all the required amino acid. Samples from each liquid culture were spread on plates containing a solution of glucose and inorganic salts, minimal medium solidified with agar. Notice the result also only the samples from the mixed culture here. notice that containing cells that gave rise the colonies on minimal medium which lacks amino acids. Conclusion, notice that because only cells that can make both arginine and tryptophan cells can grow into colonies or minimal medium, the lack of colonies on the two control plates showed that no further mutations had occurred, restoring this ability to cells of the mutant strands. Thus, each cell from the mixture that formed a colony on the minimal medium must have acquired one or more genes from cell of the other strands by genetic recombination. Okay, thanks.